Imagine walking down a street in your neighborhood. It's a completely normal, sunny afternoon.
Right, just a typical day.
Exactly. And out of nowhere, this aggressive, terrifying dog breaks off its leash and charges at you.
Oh, wow.
Yeah. You manage to get away, but your autonomic nervous system is just on absolute overdrive. So fast forward a few years, you're walking down that exact same street.
Same exact spot.
Same spot, no dog, a beautiful day. But the second your feet touch that specific patch of pavement, your heart rate spikes.
You start sweating.
Your palms sweat, you freeze. How does the biological hardware in your brain link the spatial coordinates of a perfectly safe street to an autonomic terror response and, do so instantaneously?
Well, I mean, that is the holy grail of memory research right there.
Yeah.
We have known for decades that the dorsal CA1 region of the hippocampus, the DCA1, it computes these spatial representations.
So it's basically the brain's GPS system.
Yeah, pretty much. We know the memory trace, the engram, as we call it, physically lives in these cellular networks. But historically, looking for the specific subpopulation of neurons that encoded the terror versus just the spatial map, it was like trying to isolate a single conversation at a roaring rock concert.
Which brings us to the mission of this deep dive. We are tearing into a groundbreaking paper from Nature Neuroscience that finally isolates that exact conversation.
It's a fantastic paper.
It really is. The researchers didn't just find the enneagram, they mapped its physical manifestation with this unprecedented second-by-second precision. We are literally going to look at the exact algorithms of memory allocation in your brain.
Yeah, it's mind-blowing when you get down to the cellular level.
Okay, let's unpack this. If the DCA1 is constantly firing as it maps our environment, why do certain cells get recruited to store a trauma, and what exactly are they doing at the exact millisecond the trauma occurs?
So the barrier to answering that has always been temporal resolution. For a long time, the standard method for mapping engrams was using immediate early genes.
Things like CFOS, right? Or ARC.
Exactly. CFOS is the big one. If a neuron fires rapidly, it upregulates these genes, and researchers could use that genetic expression to drive a fluorescent tag.
But the problem with CFOS is the kinetics, right? I mean, the transcription takes time, and the half-life of the protein is measured in hours. It's temporal smearing.
Temporal smearing, yeah.
So using these immediate early genes is like taking a long exposure photograph of a busy street.
Yeah.
Everything over a span of hours just gets blurred together. You can't tell who is doing what at an exact second.
Because if the shutter is open for 3 hours, you capture the cells that fired when the mouse explored the room, the cells that fired when it got shocked, and the cells that fired when it just sat there. You just cannot parse the specific behavioral epochs.
So how do we get a faster shutter speed? We needed like a microsecond shutter.
And that is the huge methodological leap in this paper. The researchers deployed an advanced optogenetic tool called f-FLiCRE.
f-FLiCRE,
yeah. It completely bypasses those slow transcriptional delays of immediate early genes by using a dual-condition protein-level tagging system. It actually requires two things to happen concurrently.
Okay, what are the two things?
The neuron must have high intracellular calcium, and the tissue must be illuminated with blue light.
Let's get into the biochemistry of that, because it's brilliant. So f-FLiCRE utilizes a truncated TeV protease, right?
Right, linked to a calcium sensor and a light oxygen voltage domain.
A LaV domain?
A LaV domain, exactly. So when a cell fires an action potential, calcium floods in. But the TeV protease remains blocked by that LaV domain.
So it's inactive until the light hits it.
Precisely. It's only when the scientists shine a blue light through a fiber optic cable into the brain that the LaV domain undergoes a conformational change.
It physically changes shape to unblock the protease.
Unblocking the protease, yeah. And then the protease cleaves a transcription factor, which travels to the nucleus to permanently tag the cell.
That gives the researchers ultimate control. They can dictate the exact millisecond the tagging window opens and closes with the laser.
While the calcium sensor ensures only the specific neurons firing in that narrow window get tagged.
I picture it as a fast flashlight being flicked on in a dark cavern. Only the bats that are actively flapping their wings at the exact millisecond the beam hits them get painted fluorescent green.
That's a great analogy. And because the kinetics of this f-FLiCRE variant are so incredibly fast, researchers could limit the tagging window to just a few seconds,
capturing incredibly brief bouts of behavior without any of that background noise. Okay, so to synthesize our methodology before we dive into the actual experiment, we're moving past the hours-long temporal smearing of older immediate early gene targets like CFOs.
Right.
Instead, we are using the rapid kinetics of f-FLiCRE, functioning as our blue light and calcium-dependent fast flashlight, to isolate precisely which neurons in the dorsal CA1 are firing at the exact millisecond a specific event happens.
Perfect summary.
So think about a memory you formed today, a specific conversation, or maybe even just reading an email. If we had that fast flashlight installed in your DCA1, what exact millisecond of neural action do you think we would have captured?
It really forces you to realize that a single memory isn't just a monolithic event. I mean, it is a temporal sequence.
Yeah, a sequence of tiny moments.
Exactly. So to understand how that sequence is stored, the scientists ran a contextual fear conditioning paradigm. We call it FC.
Okay, set the scene for the FTC paradigm.
They place a mouse in a distinct chamber, context A, and deliver a mild foot shock. The animal rapidly forms an associative memory, linking the spatial context to the aversive stimulus.
And its behavioral output is freezing.
Freezing, yes. It just stops moving defensively.
But rather than tagging that whole ordeal, they treated the fear conditioning like a 4-act play.
The 4 temporal windows.
Right. Act 1 is pre-shock. The mouse is exploring the novel chamber, gathering sensory inputs, and the DCA1 is actively generating a spatial map.
Getting the lay of the land.
Yeah. Then Act 2 is the shock. The precise fraction of a second the unconditioned stimulus is applied. Act 3 is freezing, the discrete epochs where the mouse actually exhibits that defensive posture. And Act 4 is no freezing.
Those are the periods after the shock where the mouse spontaneously resumes movement.
So they have these 4 acts.
And they took 4 separate cohorts of mice. Using the precise temporal control of f-FLiCRE, they illuminated the dorsal CA1 with blue light in only one of those specific behavioral windows per group.
So they completely isolated the cellular cast of characters for each act of the play.
They did.
And here's where the traditional models of hippocampal memory allocation start to break down for me. Logically, the DCA1 is primarily a spatial structure. It's full of place cells.
That's a classic view, yes.
Therefore, I would hypothesize that the pre-shock cells, the neurons highly active while the animal is actually exploring and mapping the geometry of context A, would form the core spatial engram required to recognize the room later.
It makes complete logical sense.
Are you saying they aren't the main memory?
The data suggests something entirely different. To figure out which of those 4 isolated subensembles actually held the functional memory, the researchers moved from tagging to optogenetic manipulation.
So they had to perform a sufficiency test.
Exactly. Along with a fluorescent marker, the f-LiCRE system was engineered to drive the expression of breaches in the tagged cells.
Okay, breaches. And for context, breaches is a red-shifted excitatory opsin.
Yeah, an ion channel that opens when exposed to specific wavelengths of light.
And they use a red-shifted variant here specifically so they can use yellow or red light to reactivate the cells later, completely avoiding any crosstalk with the blue light they used for the initial f-LiCRE tagging.
Right, you don't want to accidentally tag new cells while trying to activate the old ones. Plus, the red-shifted light penetrates deeper into the hippocampal tissue than standard channel rhodopsin.
Okay, so they have the cast tagged and loaded with breaches.
Once the cells from the 4 distinct acts were tagged, the researchers placed the mice into a novel, safe environment. Let's call it context C.
A totally neutral box. There's no shock history here.
None at all. They then pulsed the dorsal CA1 with yellow light, forcibly depolarizing only the previously tagged subensembles.
And the behavioral divergence is stunning. When they stimulated the pre-shock cells, or the no-freezing cells, the mice showed no defensive behavior.
None. They just continued exploring the safe box.
So the spatial map generated during the pre-shock exploration was insufficient to drive fear recall.
It didn't do anything. However, when they stimulated the shock cells, or the freezing cells, the mice instantly dropped into a rigid defensive freezing posture.
Instantly.
Instantly. It clearly established sufficiency. Activating those specific temporal ensembles could artificially reconstruct the behavioral output of the memory.
Just by shining a light. But a complete circuit analysis requires demonstrating necessity as well, right? I mean, they need to prove that without these cells, the memory cannot be naturally retrieved.
Right, you have to do a loss of function test. So they swap out the excitatory opsin for a highly efficient inhibitory one, ENPHR 3.0.
An inhibitory opsin.
Yeah, an ion pump that hyperpolarizes the cell, silencing it when exposed to light. So they put the mice back into the original scary context A.
And normally, context re-exposure triggers massive pattern completion in the hippocampus, and the mouse freezes.
Normally, yes. But when they pumped light into the brain to hyperpolarize and silence only the shock, or the freezing subensembles, the natural recall was severely impaired.
So the mice didn't freeze as much.
They showed significantly less freezing. And crucially, silencing the pre-shock cells had absolutely no effect on the natural memory recall. The fear response remained completely intact.
Which proves the pre-shock cells are functionally obsolete to the fear engram.
Totally obsolete.
But let's pause and look at the controls for a second, because anytime you're driving motor behavior with optogenetics, you have to prove you aren't just hitting a hardwired motor pathway, you know?
Oh, absolutely.
Like, how do we know the shock cells aren't just a generic pain reflex arc and the freezing cells aren't just generic motor arrest neurons?
The paper addresses this with two really elegant controls. For the shock cells, they used an immediate shock protocol.
What does that mean?
If you drop a mouse into a chamber and shock it instantly, before the DCA1 can form a spatial representation of the room, no associative memory is formed.
Oh, so it doesn't link the pain to the room because it hasn't seen the room yet.
Exactly. And when they tagged the active cells during an immediate shock and later reactivated them, it did not induce freezing.
Wow. So the cells only drive fear if they were recruited during a genuine associative learning event.
Yes. And for the freezing cells, they use a visual sweeping task.
A sweeping task.
Yeah, they pass an overhead shadow across the cage to mimic a predatory bird.
And the mouse instinctively freezes.
Right, but this is innate, unconditioned defensive behavior.
Yeah.
It is completely independent of a learned hippocampal context.
So it's just instinct.
Pure instinct.
Yeah.
And reactivating the DCA1 cells tagged during innate freezing produced no effect later.
That is so clean. The cells tagged in the main experiment are undeniably tied to the learned associative engram.
They are. Which brings us back to your earlier question about the pre-shock cells. What's fascinating here is why the spatial map generated right before the trauma becomes functionally irrelevant to the memory of the trauma itself.
It has to be related to the massive neuromodulatory dump that occurs the moment the foot shock is delivered, doesn't it?
Precisely. The field refers to this as US-induced remapping, unconditioned stimulus-induced remapping.
So the shock is the US.
Yeah. When the unconditioned stimulus hits, it triggers a massive influx of neuromodulators like norepinephrine from the locus coeruleus and dopamine from the ventral tegmental area.
Just a chemical flood.
A total chemical flood. And it radically alters the synaptic weights and plasticity thresholds across the dorsal CA1. The previous place cell network, the pre-shock map, is abruptly destabilized and essentially pushed offline.
It just deletes the old map.
It overrides it. In its place, a distinct, newly recruited ensemble rapidly forms a novel map of the environment, one that inherently binds the spatial coordinates to the high arousal threat state.
So the brain quite literally fires the peacetime location scouts, burns their maps, and recruits a wartime surveillance crew the second the environment becomes dangerous.
That's exactly what it does.
It's an incredible mechanism for prioritizing survival data. Okay, before we transition into the actual network dynamics of these cells, let's consolidate this four-part behavioral dissection.
Let's do it.
We established that using our rapid f-FLiCRE tagging, the engram is not a single entity. The cells active during the pre-shock window are rendered obsolete by a massive neuromodulatory reset called US-induced remapping.
Right.
And through optogenetic manipulation with breaches for activation and ENPHR 3.0 for inhibition, the data confirms that only the distinct populations of cells active during the exact moment of the shock and the periods of subsequent freezing are both sufficient and necessary to drive the recall of the traumatic memory.
Beautifully stated.
So, if you are directing this four-act play in your own brain and you experience something jarring like, say, a loud explosion outside your window, have you ever noticed how you completely forget the mundane thought you were having just seconds prior?
Oh, all the time.
Right. You might have just experienced the neuromodulatory override of US-induced remapping in real time.
It really highlights the extreme plasticity of hippocampal circuits. But the optogenetic data left a massive ambiguity.
What was the ambiguity?
Well, the shock cells and the freezing cells are both necessary and sufficient for memory recall, right? But are they the same population of neurons simply double-dipping across different temporal windows? Or are they discrete networks performing different computational roles during natural recall?
So to solve that, you can't just tag cells and play them back later. You actually have to watch the network perform in its natural state.
Exactly. And that required mounting a miniature microscope onto the mouse's skull and implanting a Drean lens, a gradient index rod lens, directly down into the dorsal CA1.
A mini-scope? That is intense.
Very intense. They utilized JG Camp 8 AIF, which is currently one of the most advanced, ultra-fast, genetically encoded calcium indicators available.
And the GCampP basically lights up green when calcium enters the cell, right?
Right. And its binding kinetics are so rapid that it can report single-action potentials with incredible temporal fidelity.
So they can see individual neurons firing in real time.
In real time, in a freely moving animal. Over multiple days, they track the exact same field of view, monitoring hundreds of individual neurons.
Tracking the exact same DCA1 neurons across days in a freely moving animal is a technical nightmare. I mean, the fact that they achieved a stable field of view to correlate the tagging phase with the recall phase is a massive testament to the lab's rigorous methodology.
It's a huge technical achievement.
So what did the calcium imaging reveal about the spatial overlap of these subensembles? Are they the same cells?
When they mapped the spatial distribution of the cells active during the 4 behavioral epochs, they found them to be almost entirely orthogonal.
Wait, really? Orthogonal?
Yeah. They are highly distinct, non-overlapping populations. The neurons that compute the unconditioned stimulus, the shock, are not the neurons that compute the defensive behavioral state, the freezing.
Which means the enneagram is a highly compartmentalized matrix. It's not just a single blob of cells.
Not at all. But the true breakthrough came when they analyzed the network dynamics during natural recall.
What happens during natural recall?
When the mouse is put back in the scary box the next day, the overall volume of calcium transients across all 4 subensembles is roughly equivalent.
So if you just looked at a bar chart of firing rates, they'd look identical.
They'd look exactly the same. But when you apply advanced statistical methods, specifically looking at the pairwise correlation coefficients between the neurons, a dramatic functional divergence appears.
Let's break down that correlation data. I look at it like a symphony orchestra, right?
Okay, I like that.
The shock cells are acting like highly independent soloists. They are individually registering specific data points of the sensory experience. But the freezing cells are a synchronized choir.
A synchronized choir?
Yes, because the high pairwise correlation implies they are part of a strongly interconnected attractor network. They are rhythmically pacing each other, likely stabilized by local inhibitory interneurons, to maintain a sustained physiological state.
The cross-correlation matrices showed exactly that. The shock cells operate with very low coactivity, firing independently, while the freezing cells exhibit this massive degree of synchronization. They fire together in tight, correlated bursts.
That mathematical distinction suggests a profound difference in network architecture.
And the behavioral data from the optogenetic stimulation perfectly supports your attractor network analogy.
How so?
Remember, when they artificially stimulated the shock cells, the mouse froze. But the instant the laser turned off, the mouse resumed normal movement.
The behavioral output was strictly time-locked to the stimulation.
Exactly. But when they stimulated the freezing cells, the synchronized choir, the mouse froze, and it remained frozen for a significant period even after the laser was completely shut off.
Oh, wow. The choir kept singing because the recurrent network dynamics had taken over.
Yes.
So the freezing cells don't just trigger the memory. Their synchronized architecture actively perpetuates the internal state of fear, maintaining that defensive posture long after the initial sensory cue has passed.
That's the power of an attractor network. Interestingly, the researchers also looked at what happens to this compartmentalized architecture over time.
Does it change?
It does. If they applied the f-FLiCRE tagging protocol during the recall phase on day 2, rather than the initial learning phase, the distinct boundaries between the subensembles began to blur.
Ah, pattern completion. During natural recall, the hippocampal network operates differently.
Yeah, the cue triggers a cascade that recruits a much more homogeneous, consolidated enneagram.
So the rigid temporal compartmentalization of the learning phase transitions into a more unified representational network as the memory stabilizes.
Exactly. The ensembles consolidate over time.
Okay, let's do our final synthesis of the calcium imaging data. By implanting GR-in lenses and tracking ultrafast JG-CampP8 calcium transients, the researchers proved that the 4 acts of memory acquisition are encoded by entirely distinct, non-overlapping neuronal populations.
Right.
Furthermore, statistical correlation matrices revealed that while shock cells fire independently as sensory soloists, the freezing cells form a highly correlated attractor network, a synchronized choir capable of sustaining the behavioral state of fear well beyond the initial stimulus.
That thumbs it up perfectly.
When you recall a strong memory, and I'm asking you, the listener, when you recall a strong memory, do you feel like a single soloist is playing in your mind, or do you feel an entire synchronized choir? Pay attention to the physical sensation. When you experience a sudden, visceral recall of a highly emotional event, do you feel a single independent thought, or do you feel a massive, synchronized physiological state taking over your breathing and your heart rate?
That feeling is the choir in your DCA1 resonating.
It's wild to think about it physically happening in your head.
It is. And if we connect this to the bigger picture, for systems neuroscience, this paper fundamentally reframes the architecture of an enneagram.
It's not a blurry blob anymore.
Right. It is not a monolithic physical trace. It is a highly dynamic, temporally compartmentalized assembly of disparate networks. Different subensembles are actively performing distinct computational tasks.
Processing acute sensory trauma versus orchestrating sustained affective state.
Exactly, all seamlessly woven together to recreate the past.
And that modular architecture opens up a fascinating and highly provocative therapeutic frontier. Right now, treatments for severe PTSD often involve broad pharmacological dampening or intense exposure therapy, which can be pretty blunt instruments.
Very blunt. They affect the whole system.
But if we now know that the shock cells govern the episodic data of the event, while a completely distinct, highly synchronized network of freezing cells orchestrates the autonomic terror, what does the future of targeted neuromodulation look like?
It's a profound question.
Could we eventually design precision therapies that disrupt the specific attractor networks of the choir, silencing the debilitating physiological terror while leaving the soloist perfectly intact? Could we allow a patient to retain the declarative memory of their past, the factual context, without suffering the autonomic response?
That would change everything.
It really would. We are just beginning to decode the algorithms of our own consciousness, and the precision of these optogenetic tools is bringing the physical reality of our memories out of the dark. Thank you for joining us on this deep dive. Next time you walk down a familiar street and feel a sudden, unexplainable rush of emotion, just remember, it isn't magic. It's a highly correlated network of cells in your dorsal CA1 singing a song they learned a long time ago.